Okay, so this video is going to be about non-ribosomal peptide synthetases. And so that's a really long word. I'm probably going to just sort it to NRPS if you ever see it like that, or just NERPs. But really what these are these large complexes that will catalyze peptide bonds, although they're not going to be catalyzing large peptides and proteins like how you think of with ribosomal translation, but they can actually accept a large number of substrates. These are mainly present in prokaryotes, such as bacteria and fungi, and they have a really high levels of diversity, and I haven't really heard of them very often. There's still a lot of research being done about identifying their structure and the kinds of things that you can do with them, but I think they're really interesting, so I wanted to explain kind of how they work, what kind of substrates, and compare them to things that we already know. So starting off, um, what are the essential domains for these NERPs peptide bond formation? So the main goal of these is that they will catalyze peptide bonds between certain substrates. And so they each have these sequences of modules. And so in this case, I'm going to be talking about them in terms of linear NERPs, but they can also be iterative or nonlinear. But linear is the one that's the most studied, and so it's going to be the one that's easiest to explain and understand. So every domain or every module is going to have a certain number of domains within them. And there are certain ones that every module has to have, and there can be some that are added to some modules and not others, depending on what your final molecule is going to be. So just to go through what every module has to have, the essential domains, you need to have an A domain. It's going to be an adenylation. What this does is it's necessary for substrate selection. It's going to activate your amino acid or your substrate that you have, and it's going to be the one that's chooses the substrate, and it's going to be the one that's going to eventually participate in peptide bond formation. So after the adenylation domain, you're going to have either the PCP or the T domain. And so the PCP is going to stand for peptidyl carrier protein, but it can also be, the T stands for the thiolation domain. And what this does is it adds your activated substrate onto the peptidyl carrier protein. So the peptidyl carrier protein is a pantotene cofactor, really similar to coenzyme A, where you're forming this thioester bond. So it's also important to have the activation using ATP because you're eventually going to be making this thioester bond, which is super high energy. So you have to have some kind of input of ATP at the beginning. So now you have this peptidyl carrier protein. This is going to be responsible for delivering your peptide to the rest of your domains. So this is what's going to kind of swing it around from the adenylation to the condensation to whatever further domains you have. So it's really carrying the protein like it says. Next, you're going to have the C domain. This is going to be the condensation domain. This is where actual peptide formation happens. So it will accept two different substrates that are already attached to the peptidyl carrier protein. And that's where the peptide bond formation is catalyzed. And this is the main one that it has, but these condensation domains are also extremely substrate specific. They're even highly stereoselective. So it really has to have condense your cargo from the T domains, and it has to be from the perfectly activated attached. And instead of a validation step like a GTP switch, it's more about substrate selectivity. And then finally, at the very end, at the C terminus, you have to have a thioesterase. And this is going to cleave the thioester bond that's connecting your peptidyl carrier protein to the final substrate that is bound. And so it's going to free the enzyme from the PCP, and that's either going to happen through hydrolysis or actually cyclization is also really common. So you can have the C terminus or one end of your protein attack the other beginning end of your protein to actually release your peptide. And so these are the ones that you have to have for peptide bond formation. You always are going to have your A, C, T, and then at the very end, your thioesterase, your TE domain. However, which allows for, there's this other high substrate diversity, which allows for other domains to be inserted as well. So for example, here this has the E domain. E is going to stand for epimerization. So you can have substrate modifications that are occurring as your molecule is even being synthesized. So in addition to epimerization domains, you can also have halogenation, oxidation, glycosylation, heterocyclization, methylation, and many more. So instead of undergoing post-translational modifications, it can occur during the overall synthesis. So that's the main structure for these linear ones, but there's also other exceptions if you have things like iterative or nonlinear. but everyone is going to have these domains to actually have your peptide being formed. Okay, so how is peptide synthesis of these non-ribosomal peptides regulated? Again, like I mentioned, these are going to be in prokaryotes. So overall, the generation of these NERP complexes is going to be, expressed in gene clusters or operons. And so each different NERP is going to be expressed all in one cluster. As you can see, these modules are all right next to each other. So they're all going to be expressed at the same time. And it's important to know that these will all be expressed based on the single NERP instead of each domain. You don't have like module 1 being expressed for multiple NERPs. It's going to be module 1, which is specific for this molecule. So since these are in gene clusters and like operons, these can actually be put onto plasmids and undergo horizontal gene transfer. So as we'll see later, a lot of these molecules are going to code for antibiotics. And so if you have this antibiotic that's being spread between bacteria because these NERPs are being expressed on plasmids that are being carried to others. And so another thing is that you don't necessarily need to make these molecules all the time, especially because there's so many of them. It's not always necessary to have these molecules constantly being produced. So it really depends on the cell's kinds of needs. So for example, in certain kinds of fungi, it's going to be dependent on your nutritional input. If you need to generate these, it can be in response to oxidative stress. So maybe these molecules will protect against reactive oxygen species, or can even be temperature regulated, since some of these can be toxins. And so maybe you have excess toxin production in response to high temperatures. So it's all really dependent on the environment that the cell's in, whether this entire operon is going to be expressed or not. And again, you're not going to have one module that's expressed while the other final one is not. They're going to be expressed all together and regulated all the same. So these aren't highly regulated complexes, especially when they're in compared to ribosomes. Ribosomes are always going to be present in the cell. They're essential for cellular function. These aren't necessarily essential for cellular function. They're going to be producing these secondary metabolites that are going to be things like virulence factors or things that just support cell function and not necessarily are essential. So the regulation is extremely tight, especially whenever you're concerning the individual modules. So like I mentioned, the C domains, those are going to be highly substrate specific for the condensation of peptide bond formation, and as well as the A domains, the adenylation domains. They're also highly substrate specific, and they're even called the gatekeepers. So each module has to be specific, and it's regulated in itself. You can't have mixing and matching of modules in natural NERP products, because these adenylation and condensation domains are so selective. So regulation is each NERP specific for each molecule, and most of these molecules will be regulated at the level of gene expression as compared to feedback inhibition or things like that. Okay, now I'm going to compare how peptide bond formation in these non-ribosomal peptide synthetases are similar and different from translation within ribosomes. And you're going to see that they're mostly similar in function and not necessarily on structure. So just kind of going through the steps, I'm going to start with initiation. So in these NERPs, you're going to have the adenylation domain. Those are highly specific, and that's what activates your substrate. In the same way, ribosomes have tRNA that also activate the substrate. One of the main differences is that the adenylation domains are based on selectivity, and your ribosomes, they have the tRNA that also have corresponding codons. And so that's kind of what determines the selectivity of your substrate, as well as the overall substrate diversity itself. Ribosomes are limited to only the proteinaceous amino acids that have L instead of D, whereas your NERP have so many substrates that are available. You can have non-proteinaceous amino acids, you can have carboxylic acids, cyclic substrates, fatty acids, methylated amino acids, and it's limited by the selectivity of your adenylation domains, but it allows for a lot of different combinations of peptides that you can make as compared to the only 20 amino acids that are dependent on the codons that you have. Okay, and then going on to actual peptide formation or the elongation step, you have ribosomes that can only make peptide bonds. Every single time you're going to have only a peptide bond that's linking one amino acid to another, whereas these NERPs, you can have different combinations of domains that will not only create peptide bonds in the condensation, but you can also have different kinds of chemical modifications like the epimerization that we saw before. So in that way, you can have chemical modifications that are occurring during synthesis, whereas you have to have post-translational modifications in the case of ribosomes, as well as the condensation domain isn't dependent on a GTP switch, where it doesn't have the same validation step that occurs in the decoding center to ensure that the tRNA has the correct codon and anticodon. It's just based on substrate specificity, which is highly selective. And then finally, the last one is going to be the termination step. The termination step is kind of the most different, where you have a thioesterase that's cleaving off the peptide carrier protein versus you have a stop codon that's going to eventually end the peptide bond formation. And so again, those are going through how similar they are in function. If you look at the structures, they're going to be extremely different. You have these different large subunits that require mRNA. There's no mRNA required in this one. There's not subunits. Instead, you have those modules that are all connected together. Usually, they're going to be linearly connected. And so it's really easier to compare this in function. But when we're talking about structure, it's going to be about fatty acid synthase. So how are NERPs similar and or different from fatty acid synthase? And so you can see even clearly that this fatty acid synthase has each individual molecule or module or domain that are present in a specific sequence that is going to be catalyzed in a specific order to create the desired molecule that you want. And they each catalyze either one reaction or one kind of modification. And at the very end, you can see that the C-terminus also has a thioesterase. And instead of a PCP, you have an ACP. These are both panthothene cofactors that are derived from coenzyme A that allow the substrate to be moved and kind of delivered to each one of these domains. So if you look at it on a linear level, they are extremely similar in structure, even though the function is going to be different. You're going to have different modification patterns in this case, where you can have maybe an epimerization domain here, but not in this one. Whereas these are going to occur the same cycle of steps every single time, which will lead to a almost symmetrical looking molecule, as well as the substrate selectivity. You have only acetyl-CoA that's using NADPH to create fatty acids, where you can have all kinds of different substrates here where they don't necessarily have to be the same thing every time. You'll have different modification patterns as compared to repeating the same functions. And then even in the name, you can see that fatty acid synthase will not use ATP. You don't have to activate anything. Whereas you have these, since you're going from a thioester of acetyl-CoA into a thioester on your acyl carrier protein, where you need that activation step, you need to use ATP, that's why it's called a synthetase, in order to create that thioester bond for your carrier protein. So overall in structure, these are very similar, even though they carry out different functions that can be compared to the ribosome. It's cool to see how certain things can be applied to other structures and other kinds of chemical syntheses. Okay, so what actually are these products? We've talked about how they're made and about the complex itself, but what about the molecules that it's actually making? And so these are going to be commonly seen in drugs, even though they're made from mainly prokaryotes. These aren't going to be really seen in eukaryotes that much. So you're going to have anti-cancer, antibiotics, antifungal, immunosuppressants. But even though you have all these things that can be converted into drugs and that will be extremely beneficial to humans, you will also have things like virulence factors and pathogenic toxins that are being expressed. So it's really what you'll notice that it's really dependent on what the cell needs. It'll be dependent upon what kind of environment the cell is in, especially for things like pathogenic bacteria, if it needs to produce toxins. But in certain ways, things like antibiotics, they can be used, especially this bacteria called Streptomyces coleoctor, and it produces this extreme variety of antibiotics. And so if we can find these molecules in the NERPs that are expressing them, we can create all this large variety of antibiotics, and that'll have a bigger range that we can use in order to combat antibiotic resistance. And besides just being able to look at different NERPs that are naturally expressed, you can actually engineer these domains to create your peptide of choice. So here, if you have each of these natural NERP systems, where each one of these is going to create a specific molecule, where they each, again, have the adenylation, condensation, those same essential domains that we talked about is going to be within each of these. In this case, the letters are just going to be corresponding to the amino acid, although you could also use a different varieties of substrates as well. If you just take the pieces that you want and you can just fuse them together into another linear structure, what you'll have is this will express a artificial peptide that you want that can serve a specific function that you need. And so it's a little bit, again, the same thing can be done with ribosomes by creating your own kind of changes to mRNA. But this allows you more diversity with better substrates. You can have chemical modifications along the way instead of worrying about trying to add those chemical modifications post-translationally. And so there's been quite a bit of success with this, although there has been a little bit of trouble since the condensation domains are so highly specific. You need to have a condensation domain that'll allow for each of these substrates to be put together. So you can have the conserved adenylation domain, but the condensation domain has definitely been something that struggle that has been need to overcome. But overall, this shows a lot of promise that by putting these specific domains or modules in different orders, you can create different products that can serve all of these different functions that can especially be used for things like drug development that are beneficial to humans that we can use just by looking at these secondary metabolite products that are made in these non-ribosomal peptide synthetases. And so we know how they're regulated, we know how they overall work with the domains. They're necessarily simple and they have the same kind of properties that ribosomes and fatty acid synthase do. And so just by taking these and modifying them into how we want them, engineering them based on the domains, and these can go as long, as simple or as complex as you need them to. But they're overall fairly efficient at creating these secondary metabolite products if you're looking at their overall significance, especially in designing or finding different antibiotics or anti-cancer drugs. So I think this was a really cool topic, and I'd never really heard about it before. So I hope that you kind of learn and understand something, and I hope you enjoyed this as much as I did. So thank you.